This slide shows an older idea about how GC1F protein might be changed into GcMAF. This idea was first reported in 1993 by Dr. Yamamoto and Dr. Holi from Tokushima University. They suggested that when the body has inflammation, certain sugars on the GC1F protein are cut off. This happens with the help of two enzymes, one from B cells, called beta-galactosidase, and another from T cells, called sialidase. Step by step, the protein is modified, and in the end, it can be turned into GcMAF. Next, I will talk about the classic Nagalase theory. This theory was first suggested in earlier studies. Nagalase, also called alpha-N-acetyl-galactosaminidase, is an enzyme that can be released by cancer cells, virus-infected cells, and even some normal cells. In cancer patients, higher levels of Nagalase have been found in the blood. Nagalase works on the vitamin D3 binding protein, also called GC protein, by removing its sugar parts. Once these sugars are gone, the GC protein can no longer be turned into GcMAF. Without GcMAF, the immune system becomes weaker. According to this theory, this process is a key way that Nagalase lowers immune activity. The idea has been widely discussed, although it is still debated. There are two types of Nagalase: exotype and endotype. Exotype Nagalase is released not only by cancer cells and virus-infected cells, but also by normal healthy cells. Endotype Nagalase, on the other hand, is released by cancer cells and virus-infected cells, but not by normal cells. The classic theory focused mainly on the endotype form. However, in most laboratories, only the exotype form is measured. Over 30 years ago, it was very difficult to measure endotype Nagalase directly. To solve this problem, Dr. Uto from Tokushima University created a special artificial substrate for exotype Nagalase. This method made it possible to measure exotype levels in the blood of patients. So, what is the difference between exotype and endotype Nagalase when it comes to breaking down sugars? First, GC protein has a more complex sugar structure made of beta-galactose, GalNAc, N-acetylgalactosamine, and threonine. In contrast, GcMAF has a simpler structure with just GalNAc and threonine. Second, the two forms of Nagalase behave differently. Endotype Nagalase can break down both GC protein and GcMAF. Exotype Nagalase, however, can only break down GcMAF, not the more complex GC protein. This slide shows the activity of GcMAF made from the blood of cancer patients, based on experiments carried out at Tokushima University. The graph demonstrates that GcMAF from cancer patients' blood is almost as active as GcMAF from healthy individuals. This means that even in cancer patients, their own GC protein can still be converted into active GcMAF. This finding contradicts the classic Nagalase theory, which suggested that GC protein from cancer patients could not be turned into GcMAF. It is also worth noting that only a very small amount of GcMAF was used in this experiment, and clinical data suggest that Nagalase may act more as an inflammation marker rather than a strict blocker of GcMAF production.